I work as a computer scientist at the University of Miami, where I do research on cancer transcriptomes and metagenomics. And I used to work with Jennifer when she was down in U of M. Then last year I went back to finish my PhD in computer science at Florida International, and I work there developing analytical tools for the research in transcriptomes and metagenomics. And today I'll be talking a little bit about genomics and some of the work that we do with Jennifer and some of the work that we've done in the past. And I'll give you guys a brief overview of sequencing technologies so you guys can get familiar with the type of data that we use. I'll talk a little bit about a particular protocol of sequencing, which is called RNA-Seq. And then I'll talk a little bit about the challenges that you would run into when you're doing an analysis. And then hopefully I'll have some time to kind of go over the visualization aspect of it, because we generate a lot of data and then we try to make sense of it and visualize it. So sequencing technologies. So DNA sequencing, like what we know it, is basically getting the primary structure of a molecule. And we're interested in getting the A, T, G, and C of a particular DNA molecule. These are some of the definitions I got from Wikipedia. What we basically want is like a ticker tape of letters. They're 4-character strings, A, T, G, and the combination of them results in a sequence that we're going to analyze. So up until a couple of years ago, most of the sequencing was done by a method called Sanger sequencing. It was developed in 1977. And I'll be talking a little bit about that method and what the differences is to some of the more modern technologies. What we do a lot of work in is in the basis of a genome, or in the context of a genome. And we primarily work with the human genome when we're doing cancer transcriptomes, or we work with a lot of genomes when we're doing bacterial metagenomics. When we're working with the human genome, we only work with one genome, and that's a human. And that's a consensus human genome that was originally sequenced in 2001. For the bacterial genomes, we use a database from NCBI. We use a GenBank database. It contains about 34,000 bacterial genomes of different sizes. Bacterial genomes go from about 10 to the 3rd to 10 to the 6th in size. So they're much smaller than, say, the human genome, which is about 3.1 billion characters. And the bacterial genomes that we get is basically from the NCBI database. We did a project about 3 years ago. And when we did that project, the size of the database was about 5,000 genomes. And now, if you go check it today, it's about 34,000. And the genomes are in many stages of finishing. Some are complete, some have gaps, and some are just a bunch of, they call it contigs, just long sequences that you could stitch together into a scaffold and eventually call it a complete genome. So some history. These are some of the landmark years in sequencing. You have 1977 when you have Sanger sequencing, and then in 2001 the first draft of the human genome was published, and then in 2007 you start getting these next generation technologies. You start getting this high-throughput sequencing. Some of the first data sets contain a couple of million reads, and now we're getting billions of them. And so Sanger sequencing is what's usually called the gold standard of sequencing. It was done by hand. It was very time and cost, and the costs were very high. But the advantage is that the sequences were relatively long, about 800 to 1,000 bases, and the quality was really, really good. It contained almost no errors. But the disadvantage is that it took a long time to generate a genome. So the human genome that was published in 2001 was mostly done with a variant of Sanger sequencing, and it cost billions of dollars, and it took over 10 years. Now you can reasonably sequence a human genome in about a week with about $10,000 worth of consumables. That doesn't take into account the analysis, but it's just what you're going to burn at the laboratory. So now you have these so-called next-generation sequencing technologies, and what characterizes them is the amount of sequence that you get per unit time. So now we get a lot of sequences in seconds, and these technologies, there's many vendors. There's Illumina, there's Pacific Biosciences, Oxford Nanopore, and what they're all characterized with is that they all parallelize the reactions to get the sequence, and they all parallelize it in a small solid surface, which looks about. If you ever messed around with microscopes when you were in high school, the flow cell looks about the same. And what happens in the little flow cell is that all these sequencing reactions take place at the same time, and you have some type of camera that captures it, and it's able to give you the sequence at that moment in time. So you take a lot of snapshots, and then you start building the sequence. But you're able to do this in parallel. We get a lot of data, and the costs are relatively reduced as opposed to Sanger sequencing. And as I said before, in 2007, we were getting millions of sequence reads. Now we're getting billions. And they're all relatively cheap to generate. Each sequencing instrument is about the size of this podium, but the new ones are even smaller. And you can get some benchtop sequencers, which are about the size of a small printer, that they don't have the throughput that these machines have, but they can do small genomes in a couple of days or in a couple of hours. So it mostly depends on the application that you're using it for. So that's the picture of one of Illumina's machines. I'm going to be talking about Illumina most of the time. I'm not endorsing Illumina; it's just what we use. I'm not a stockholder, so you can use anything. But it's among the cheapest. And in the beginning, the reads, so the sequences that it was generating, were not that long. They were about 32 bases. Now you can reasonably get sequences of about 150 to 300 bases long. And the quality is really, really good, about, I think, 99.8%. Some of the other instruments, like Pacific Biosciences, they can do longer reads, but at a cost of accuracy. So they have accuracy rates of about 70%, or 80% in some cases. And what people do if you're building a new genome is that you would get the longer sequence and then kind of patch it up and correct it with the shorter sequences. And so when you're dealing with sequencing, especially when you're doing human sequencing, you're always going to be hearing or listening sequence coverage. And that's the sequence coverage is measured by an X at the end, which is like 28X or 30X, and that tells you how deep your coverage is. And what that basically tells you is that for every character in the sequence, the X tells you how many reads map to it. So no, I don't have, I thought I had it. Yeah, here. So that little stack of reads is basically telling you the X of coverage, and that tells you how good of a coverage you have for a particular region. So over here we're looking at a gene, and you look at the genome. I can't move. But at the bottom, that's the reference genome. And then at the top, we have our experimental data, and that's with the data from the machine. And each little read is a little sequence read that we got back from the machine, and they're all going to map to a certain position. And that little stack is your coverage, and the higher the coverage, the better. Because if you're calling variants or mutations, the higher the coverage, the better. But it's also costlier. Huh? Oh, sorry. Yeah, so some of the commercial platforms that are out there, like I said before, Illumina, Pacific Biosciences, Life, Roche. The new one in this slide is Oxford Nanopore, where what they're trying to do is they're trying to create a little small pore and then get a molecule of DNA and then ratchet it down down that little hole, and then by measuring an electric potential across each base, they're able to tell, oh, this is an A, this is a T, this is a G, or this is a C, and then you just kind of ratchet it down, and you start getting like a ticker tape of an actual read. And those are what's called as third generation sequencing. So all these commercial platforms, what they have in common is the number of parallel reactions that they're running. The number of sequences that they output is also kind of different. So Oxford Pacific Biosciences, they can do long reads, but the amount of throughput, the amount of sequence reads that they generate are not that high, as opposed to say like an Illumina machine, which generates just humongous amounts of data. And then it's a trade-off. One's higher quality, the other one's longer, lesser quality. So this is just to give you an example of what this machine can generate. It can generate 120 gigabases every 27 hours, and that translates reasonably to one human genome at 30x coverage in a little bit over a day. You have 1% to 2% error rates, depending on the sequence chemistries that you're using. It has an onboard computer, and it can generate about 32 terabytes worth of data alone, because it's capturing images. You don't need to keep these images at the end. It's just not feasible because they're so big. So what usually happens is so you capture the images and then you discard them. And the workflow, you prepare your library, so you go out, you take a sample of the RNA that you're going to be analyzing. Then you generate a cluster, which just means preparing the DNA and the RNA that's going to go into the flow cell. Then you do the sequencing, which is where you actually take the pictures. Then you do image processing, where you infer the bases. You discard the images, and then the fun starts. That's when the data analysis finally starts. So again, you can expect about 32 terabytes worth of image data. That's for one sample and one run alone. And at the end of the day, after you have to process all this data, for each sample, you usually have 250 gigabytes worth of data. To give you an idea of how big that is, my little MacBook over here has 256 gigabytes worth of space. So I wouldn't even be able to fit a sample of all the reads in my computer. I wouldn't have the space because the operating system takes about 10 gigabytes. So there's no way. So we generate all these reads, and we have billions of them. And well, what do you do with them? What can we do with it? And one of the cool things that we do is RNA sequencing, where we actually don't sequence the DNA, but we sequence the RNA. And these sequencing machines, they're DNA sequencers. So they don't take RNA. So what we have to do is when you capture the RNA, you have to convert it to DNA, which is cDNA, complementary DNA, and then you sequence that. And so what's RNA? So RNA is so if we look at the typical textbook example of what the workflow is for transforming a DNA into an actual protein, you go through DNA, through RNA, and through protein. Now, this is not really accurate because you can also go from RNA to DNA, but this is the basic workflow. Now, why would you want to capture RNA? It's because RNA carries the message of the genes. So if you wanted to study what your transcriptome looks like right before lunch and after lunch, you could just sample the RNA, and then that's going to give you a reasonable idea of what your stomach or what your body was like before and after lunch. And the transcriptome, what it is, it's a set of all RNA molecules in a cell. So it's a cell. It's an entire set of genes, and not necessarily classical genes, but just genes that are being expressed. So RNA transcripts that are being expressed at that particular time. And RNA-Seq is just the application of NGS to take a snapshot of the transcriptome at that moment in time. And so again, why would you want to do transcriptome analysis? Because, well, it gives you a catalog of all the RNA species that are happening in a particular time. A popular one is that you can accurately quantitate gene expression. So before the advent of RNA-Seq, if you wanted to study gene expression, what you would do is you would run a microarray experiment. And you would get a microarray chip, and then you would get your RNA sample, then you would wash it, and then you would analyze it. And if you've ever worked with bioconductor, there's a lot of tools in there for analyzing microarray data. The problem with microarray data is that you would only be able to detect whatever was on the chip. So if that chip was designed for 20,000 genes, then you would only be able to study those 20,000 genes. And one of the promises of RNA-Seq is that not only you can study those 20,000 genes, but you can also study new genes that have not been analyzed, that have not been cataloged. And so once you start applying these type of workflows to RNA data, you can start potentially discovering new species of RNA. There's a whole family called non-coding RNAs. When you think of a gene, there's about only 20,000 genes that create a protein, but you also have all these non-coding entities that people call dark matter that are in the genome and that are actively producing things. And it's kind of hard to see what exactly they're doing. And with RNA-Seq, you're able to capture all that information. And so with gene expression, well, you can study all kinds of things. You can identify viral infections in a cell. Sometimes viruses insert themselves into the genome, and if they're expressing a particular protein, then you can capture that with RNA-Seq. And you can also find all kinds of susceptibility to cancer. I work with melanoma samples down in Miami, and we use a resource called the Cancer Genome Atlas. They have a lot of samples for many types of cancers, and so if we study gene expression, we're trying to get an idea of what pathways and what things are activated in a particular set of samples. And RNA-Seq basically is a digital count. One thing that people say is that before, when you were doing Sanger sequencing, you were doing it was analog technology. And now with next generation sequencing and RNA-Seq, you're getting digital counts. You're actually getting pieces of information that were present in your sample when you captured it. And so before with microarray data, you would measure the intensity of an image, and that intensity was you could take it as a proxy of the expression of a gene. But now with RNA-Seq, we actually get a digital count, a little piece of information of actual biological information that was present. And so with RNA-Seq, what you want to do at the end is basically create a pie chart that gives you the distribution of the expression of all the genes. And you can create these monolithic pie charts that give you the distribution of all the genes, or you can take individual ones for a particular for a given gene, for a particular one. And you basically have two types of projects. In a mapping project, you start working from a known reference. So you take the known reference of the human genome, and then you go and you take you run your RNA-Seq experiment, and one of the first things you do is, okay, I have all these little reads, the guys in black and blue, and what I want to find out is their position in the genome. And you go and you use an aligner, and then that's going to give you the position of the genome. Then you know what the coordinates of the gene were. So then you can say, oh, this particular gene has 15 reads attached to it. So the expression of this gene is going to be 15 in a given data set, in a given condition. Another type of project is a de novo, where you don't know exactly what you're going to be studying, and you don't have a reference sequence to align to. So you take all those little reads and you start seeing how they overlap and see what kind of scaffold they create. And then this is what the data looks like when you visualize it in a genome viewer. So the track at the bottom is a gene, And then if you zoom into it, we look at the window at the top. The little guys in red and blue are the reads, and then you see that little peak in gray at the top. That's a coverage for that particular region of the genome. And the high peaks just say that it's a lot of reads that are mapping there, a lot of sequence coverage there. And usually the peaks are going to correlate to exons, and then the thin blue line is an intron. And what that tells you is that there shouldn't be any expression there, because you're when gene expression, the only things that are really expressed are exons. So when you have an intron, that region with the thin blue line, nothing should be in there theoretically. But because of these dark matter expression and alternative splicing, you get a lot of things that are in there. So what are the challenges? So we've been talking about big data, but in sequencing, what's actually happening is that we have small data. The data sets have gotten big, but the actual data points have gotten smaller, more accurate. So we have smaller data that's more accurate and more precise, but we just have a lot of it. And sequencing now allows you to ask all these questions that we didn't have before. So over here, you're looking at mapping the hallmarks of the lung cancer and the visualizations that they have. It's really cool because you can see the areas that theoretically nothing should be mapping to, nothing should be expressed, and you can see these dark matter areas that are popping up like hotspots, and something's going on in there, and we don't know what. And so another challenge is that the pace of sequencing is outpacing our ability to analyze it. So over here we have a popular image from NIH. Across we have all the years, and then you see the cost of the genome and Moore's law over at the top. And what this graph basically tells you is that producing sequence information is rapidly outpacing our ability to analyze it. Now and Moore's law is not keeping up with it, because ideally both will be in sync. So now we're just generating all this, just tons and tons of data, and we don't know what to do with it, or even where to keep it. And the cost per genome over here, you can see it goes down. Now it's still not hovering right about $1,000. I think there was an X prize to whoever could generate a genome for under $1,000. And there's a couple of companies that if you squint hard enough and if you do a lot of financial gymnastics, they're able to claim, a sub 1,000 genome, but it's still not feasible. And then that's only generating the data. What happens afterwards? That's a real cost. All right, and one thing that I should say, that this is the cost of resequencing a genome. So for the human genome, it would be, okay, we already have a reference that we generated a couple of years ago. Now we're going to go and resequence it. Now it's going to be relatively cheaper. And so we work with a couple of big data sets. These are two that we've been working on with Jennifer. The first one is the Cancer Genome Atlas, which I mentioned before, and the second one is the Human Microbiome Project. The Human Microbiome Project, or HMP, what they're trying to do is catalog the microbiome at different parts of the body. So they're trying to look at the bacterial titer that say your stomach or your ears or your nails or all those body sites have, and they're trying to create a profile of all those bacteria there. These are normal tissues. This was for phase 1. I think phase 2 is actually going to have treatments for those sites also. So we're hoping on getting samples where you can see the bacterial population at one point in time in normal tissue, and then we can study it again in treatment tissues at the same site. They have about, I think, 1,200 to 1,900 samples. When we analyzed it in the last paper that we did, we analyzed 2 samples. It took about 6 days of cluster time and generated about 4 terabytes worth of output data. And that was just for 2 samples. So with these new sequencing technologies, we have these base resolution expression profiles. And these are great because you can look at it at a very, very high resolution what the expression value is, but the drawback is that it's a lot of data, and keeping it around is a headache. We have to stay ahead of the HPC administrators and kind of keep moving around data and hiding it from them, because if not, it gets deleted after 21 days. And Joel, if you're looking at this, then I didn't say anything. Right, so we generate a lot of data, and most of the time it's easier to just store the scripts and the software that generates the data, and it's not feasible to store the results because they're so big. So most of the time we generate the data, we get a summary of it, we store it, and then we delete the output. And if we need it again, it's just cheaper to rerun it, spend another week of cluster time than it is to actually keep it in cold storage or try to buy the storage. And so this is some of the pipelines that we use. We basically go from left to right, and it's from it's basically capturing the data on the left and then going through all the process of analyzing it and then creating the reports at the end in red.
Camilo?
Yes.
So are you going to talk about how you turn that huge amount of data into something smaller? Because can you run an analytic process on it and then have something smaller to hang on to use in the future?
No, I wasn't going to talk about it, but I'd love to. Okay. So yeah, when you first run the analysis, the data files are huge. There's no way we can analyze them. It doesn't fit in memory. So what we do, there's a lot of tools that compress the data into a binary format. So when the data comes out, it comes out as human readable strings. You look at a nice report of each read of each sequence, and those reports are nice, but each file contains millions of rows. There's just no way of actually doing anything with it. So what we have to do is we have to turn that data into binary formats, into ones and zeros. Once we have that, in most cases, we're able to generate what's called a BAM file. It's a binary representation of the file, and that's hugely compressed. Imagine keeping around zip files of this. So all the output files that we have is character data. So when we compress it, it's a very efficient compression. Most of the tools are designed to use compressed data, and now we're starting to see some tools that don't even require an actual data file to be present in your file system. Some of the new tools, what they do is that, okay, we're going to start analyzing the data and we're going to start to stream it, and we're just going to have another utility downstream that's going to capture the stream of data, and it's going to analyze it in real time. And as it's analyzing, it's going to start building a report, and at the end, it's going to dump out the final report. But then there's no large footprint of data that you have. But right now, almost all the tools, in fact, almost all the stages in there, there's a second green one that's a SAM to BAM. That's a step that we compress the data, because the data is it's too big. Right, so it's a data deluge. We're getting inundated with data. These new tools that allow you to do streaming analysis are really nice because you don't need this huge hard drives or huge storage to maintain the intermediate data. And some of the tools that we've been using in the past have gained that capability, like Bowtie or Toppat. They can do streaming analysis, which is really nice. And so at the end, what we try to do is visualize the data. When we're trying to create a visualization, we try to keep things simple. There's this magic number 7 ± 2, and it's from a paper that came out, I think, in Psychology a couple of years ago, that humans are able to yeah, so I'm not that familiar with the paper, so if I butcher the reference, I apologize. But we try to keep the visualization simple, with not too many complex things going around. And the most important thing that we talk about is what's the job to be done for this visualization. What are we trying to do? What are we trying to show? What are the important features and patterns? And where are we going to be displaying this? Are we going to put this on a paper? Are we going to put it on the web? Or how are we going to use it? This is a visualization that we did for a paper that we published in January, I think. It's a frequentist. Yep. And so what we are showing here is the small image shows you the p-values for all the bacteria, for all the bacterial genomes that we are analyzing. And then the big guy, what it tells you is that we run many simulations at different mutation parameters. And the right side of the diagrams are the low mutations, and then the left side of the diagram are the high mutations. And then you can see how things are popping in and out on the diagram. The circular diagrams are created with an application that's very popular in bioinformatics called Circles. It's a Perl script that takes about a week to set up on a regular computer. It's a real headache, but it does pay off at the end. We also use R and ggplot2, Python, MATLib, and we also create pipelines with KNIME. I'm not sure if you're familiar with KNIME. It's a nice pipelining tool that's open source based on Java, and it allows you to create these nodes and then visualizations at the end. These are some resources that are included there, if anybody wants to check them out. The one at the bottom, Visual Statistics, it's not a typo. That's how they spell it. And it's really fun. Yeah, so thank you. So any questions, comments, queries? Yes.
How close are we to finding a way to solve the problem that you have found with the genetic marker?
Right. So that's very hard because right now we can describe the problem, but taking an action from that problem, it's still I would say a couple of years away.
So my question is, there are techniques for sequencing where you can get information that is strand specific, right? And I was wondering, is that a consequence of the library, how you do the library prep, or is that a consequence of the chemistries that you're using? Like, how do they get the strand specificity from the sequencing?
It's a mixture of both. So when you do your library prep, I don't know if you have this step. Yeah, here. So when you're in step 1 in your library prep, the preparation protocol is designed for strand specific, for being strand specific, and also the chemistry that you're using is also tuned for the specificity. So what you end up getting is reads that look like this. So the blue guys are going one way, and then the red arrows are going another way. If you weren't using strand specific, then probably all of them would be facing one way. So it's a little bit of both. Yes.
I have a question. So you mentioned next generation sequencing, right? So the one before that, for example, the FM matrix 500K. So does that mean having the next generation sequencing, we don't need the 500K? And so by having more in terms of SNP or data, So minor allele, like those kind of, you can now type the minor allele frequency really low one, right? So, yeah. But we don't have the ability to analyze those data. So does that mean I just want to have an understanding by having this tool, how to use this tool, or yeah, which one to choose from? Yeah.
It all comes down to economics. The Affymetrix one is considerably cheaper than running an RNA-Seq experiment. An RNA-Seq experiment, I think nowadays goes for $1,300 if you do it in-house, probably a little bit less. If you run an Affy chip, you're looking at $200, $300 per chip.
Yeah.
So if you have the budget and if you want to run multiple samples, running a sample at, say, $1,000 compared to running a sample at $300, that's going to factor in your decision. They're not got my tongue twisted. They're not exclusive. You're going to choose. It's mutually exclusive. Sorry. You're going to pick whatever's in your budget. If you have the budget for a larger analysis and you can actually afford it, then sure. I mean, get the best bang for your buck. But if you're in a budget, Affymetrix still gives you plenty of useful information. So it all depends.
Thank you.
Sure.
So one of my other questions was, one of the other differences between the Affy chips and the RNA-Seq is that the RNA-Seq data is count data, essentially, right? And the Affymetrix data is not count. It's continuous. It's an intensity, right? Yeah. Yeah. So when you're working with the NGS data, the statistical distributional assumptions are different.
Oh, yeah.
Because the data's really count. So it's sort of, first it was sort of, people thought, well, it's negative binomial, and then it was negative binomial Poisson, and then it was negative binomial Poisson, zero inflated, and then it was, so there's, this whole process going on in the literature of what is the distribution of this data, because it's count, and it can be different for every gene. Yeah. It's not necessarily the same, so. Yeah,
and we've been exploring algorithms for doing this, for trying to figure out what kind of distribution is the best one. And the answer that we came up with is that there's none. Because if you take one data set, then you're going to get one type of distribution. And then you say, oh, it's a negative binomial. Then we're going to go and we're going to analyze everything with that assumption. And then you stumble across a data set where, no, it's not negative binomial. And then we have 5 different methods, and we run 5 different methods, and we get 10 different answers. So yeah, I don't think there's there's no one distribution to rule them all, so to speak.
And then, unless you're doing single cell sequencing, most of the tissues that you're doing sequencing off of are actually a mixture of different cell types. Yeah. All right. Yeah. So some people have looked at it from the perspective of doing a mixture of distributions. If you could sort of estimate, or you knew the proportions of the different cell types in your tissue, you could try to deconvolve the count data, the signals, into a mixture of the different component. Yeah. Yeah. I mean, it just makes it more complicated, but. Yeah. Okay, I have one more. See, I get to answer all the questions. I get to ask all the questions. Okay. How does this differ? And I don't know if you know this, but how does this approach differ? How does RNA-Seq differ from, say, ChIP-seq or HiSeq data, right? Which measure different things, but
they definitely measure different things. The preparation protocols are a little different, say from ChIP-seq to RNA-Seq. At the end of the day, you have this machine, and that machine is gonna sequence DNA. So you have to go through a lot of transformations of your library, as far as to, okay, we have ChIP-seq data, which measures the methylation sites across the genome. And how can you turn that data into something that the machine can read and then can use? It's all in how you prepare the library and how you capture that information. At the end of the day, you're doing DNA sequencing. So you're going to go through a couple of steps that you're going to get your RNA or your ChIP-seq, and you're going to fiddle with it and then give it to the machine.